evening, everybody, and dear colleagues, welcome to this session on the red coral. It's really a great honor to moderate this session, and first I would like to thank the CIESM committee and particularly Dr. Laura Giuliano for their effort and support in making this session possible. We are going to have scientific presentations, and then we will have a film. And I have the honor of starting with a presentation on behalf of my colleagues from the Centre Scientifique de Monaco and the CIESM Channel Consortium. So the title of my presentation is Advancing Understanding and Conservation of the Mediterranean Red Coral, a Comprehensive Initiative. So I'm going to present you this research program, this research program on Corallium rubrum, the Mediterranean red coral, which is a collaboration between the Centre Scientifique de Monaco and la Maison Chanel. This program is developed within the framework of the Unité de recherche sur la biologie des coraux précieux, which in English would be the Research Unit on Precious Corals. And we have a steering committee which is chaired by Her Royal Highness la Princesse de Hanovre. And this committee also consists of members from the Centre Scientifique de Monaco, from members from the Maison Chanel, and from international scientists. And this committee meets annually to look at the progress of our work. The unit is located at the Centre Scientifique de Monaco on the harbor of Monaco, and it occupies 2 floors that are outlined on the picture. The Centre Scientifique de Monaco, just to present it to you, was founded in 1960 by Prince Régnier III, and it's the research institute of the Principality of Monaco. It is dedicated to both fundamental and applied research, and it consists of 3 departments: the medical biology department, the polar biology department, and the marine department. The marine biology department specializes in the study of corals and coral reefs, both from tropical, temperate, and also deep sea zones. Our research spans from the level of the gene to the reef, and we also integrate socio-economic aspects. The scientific team working within the CIESM Channel Consortium belongs to the 2 teams of the Centre Scientifique, the Coral Physiology team and the Coral Ecophysiology teams. And in this unit, we have 6 people who are dedicated only to the program on the red coral. The others also work on the tropical corals and the deep sea corals. The entire consortium is led by Professor Denis Allemand. And as you can see, among the scientists, we have 5 divers, and you will understand after how important they are for this program. So now I will give you some information on the red coral. Just I imagine that you all know that this is an animal, and this is a colonial animal, meaning that it is made of individual units which are interconnected together. And this animal elaborates a skeleton. The skeleton is of calcium carbonate, and it also contains organic molecules, and that's why we call it a biomineral. And this biomineral, due to its beautiful red color, has been used since ancient times for art and jewelry. This coral is distributed in the Eastern Atlantic, in the Mediterranean, and in the Adriatic. We can find it from 10-meter depth until 1,000-meter depth. And it doesn't need light, so we can find it suspended on the ceiling of underwater caves. The red coral plays a crucial role as a key component of the coralligenous, habitats, which are the temperate equivalent, we can say, of the tropical coral reefs. These habitats, they host a rich biodiversity of species, including algae, gorgonians, sponges, and various invertebrates. So these are really vital ecosystems for maintaining biodiversity. So this coral, unfortunately, has been harvested because of its importance for trade, and there has been illegal and unsustainable harvesting. And also, as many other invertebrates, it is facing environmental change, such as rising temperature of increasing ocean acidification. And for these reasons, it has been listed on the red list of the International Union for Conservation of Nature. And also because it has a very slow growth rate of about a few millimeters per year, we need really initiative for the conservation of this species and its sustainable management. And that is where our research program comes into play. And so now I will present you the 3 work packages that are within this research program. Work package 1 is about fundamental research, and it is aiming at studying the complex biology of Corallium rubrum. The work package 2 is applied research, and it is to find solutions for sustainable management of this coral. And finally, the work package 3 is about raising awareness for coral conservation. Work package 1 has 3 different axes. One is growth and calcification, the second is skeleton color, and the third is microbiome and coral health. Concerning growth and calcification, we are wondering what are the molecules, the biological mechanisms that are underlying the formation of the red coral skeleton, and if we can boost this process. And we are looking at different factors, both environmental factors and intrinsic factors. And we are also looking at different scales. We are looking at genome diversity, we are looking at gene expression and skeletal proteins. And as you can see on the right, we have already obtained nice results that have been published or that are under publication. I wanted also to mention an advanced technique that we have developed and which consists of injecting molecules, which are molecules of interest for boosting coral growth directly in the coral. And you can see there that it's something which is quite very tricky because we have to insert the syringe in the coral, which is very small, but we have succeeded and we are now having the first result of this process. I say that the second axis of work package 1 is skeleton color. And there we are wondering what are the molecules which are responsible for the coral, the red coral skeleton color, and whether this has an influence on coral health and coral growth. And as you can see on the representation, we have already identified a few years ago that the color is at least partly or mostly due to a pigment, a carotenoid. And we know that the carotenoid cannot be synthesized by the animal. It must come from another source, and we are just wondering what is this source, whether it is some things like the food or maybe microorganisms. And then we are also wondering what are the pathways for this pigment to go from the source to the skeleton. Then the third axis of this work package 1 is microbiome and coral health. And we are wondering what are the microorganisms, I mean bacteria, fungi, protists, and viruses that make up this holobiont. In other words, we want to identify the composition of the coral microbiome, and similar to humans, we are wondering what is their role in the health, in the nutrition and development of the coral. Then we have this work package 2, and this one focuses on culture of the coral both in the field and in the laboratory. Our goal is to determine the best conditions that will promote coral growth and reproduction. And we are particularly interested in the composition of the substrate, the quality of the substrate, and the sex ratio of the colonies. And you will hear talks about this after. And for this program in situ of the coralliculture, we have collaborators from the local laboratory. And this laboratory is a scientific multidisciplinary group aiming to deepen our understanding of the dynamics of benthic environments in order to contribute to the creation of a solid scientific basis for future marine spatial planning. This laboratory is located in Bagnols-sur-Mer, and this program is also partly funded by the Prince Albert II Foundation of Monaco Foundation. The third part of this program is the work package 3, and it's about raising awareness of the public. And we have different actions. We go to school and we do educational conferences. We also have published a book which name is Le Mystère du Corail Rouge, Red Coral Mystery, where we can see a very nice hand-drawn illustration. And this book is presented to different classes in schools across Monaco. So this was the presentation of the research program, and so now you will listen to talks. The first one will be by Lorenzo Bramanti from Le Cob Laboratory, and it will be about in situ coralliculture in Bagnols. So please, Lorenzo.
Okay, I think I can start. Good afternoon to everyone. Thank you, Sylvie, for the introduction. I'm going to present the cooperation between CNRS, that is the institution for which I work, and the Centre Scientifique de Monaco. And I will start from where Sylvie stopped. So we have a species of high in economical value with extremely slow growth rates, which is subject to disturbance, both anthropogenic and environmental, and for which a lot of conservation management plans are ongoing. So it's the classical characteristic for a species that, for which restoration can be planned. But when we speak about restoration, we speak about a science developed in a tropical environment on species with relatively fast growth rates and different life cycle with respect to the Mediterranean one. And the same scientists, when dealing with science, are a particular approach. So if in ecology conservation and management, the approach is based on observation, experimental design, hypothesis testing, and statistics, when dealing with transplantation, scientists looks like they lose their scientific approach and they transplant compulsively, waiting to see what's going to happen. So what we propose, we think that is necessary is a scientific approach to restoration and transplantation. Mainly, knowing the characteristic, the life cycle characteristic of the species can be useful. For example, the characteristic of corals to produce thousands of larvae with a very low success to adultness suggests that acting in this part of the life cycle can, in increasing the success here, can have an outcome of more than thousands of colonies transplanted. And for red coral, Mediterranean red coral, the situation is even more particular because it's characterized by gonochoric species, so male and females are separated, and there's internal fertilization. So to work on the transplantation and the restoration of these species, we need a scientific approach. We need to know the characteristic of the life cycle. But to do this, we will need a high scale because we need to do it in natural environment. But in natural environment, we cannot control parameters to do a scientific approach. So if we cannot bring the natural environment in the laboratory, we decide to bring the laboratory underwater. This is the Hotel Coral, which is a concrete 1 meter cube cave that is meant to study, to apply a scientific approach to study of restoration. It was inspired by the seminal work of Denis Allemann that in the 90s put big concrete caves in the waters of Monaco. I was a student at that time, and I always dreamed to do something like that. But I needed something different, so I need an underwater laboratory for hypothesis testing. And this is why the vault of this hotel coral are made of sliding tiles on which you can attach one or more red coral colonies, and then control the experimental parameters like sex ratio, density, and distance by keeping them in a natural condition. More than a coral hotel, in fact, it's a coral resort, because we have 6 caves put at 40 meters depth in Monaco and in Banyuls in a shape of a hexagon with known distances, which allow us to have an artificial population inside. So we know everything about the individual of the population inside the caves. We know details on the morphology, on sex determination, and on genetic characteristic. And this allows us to close the cycle, and we can have mothers that release larvae, and we have coral babies. And this is, for example, probably the first ever video of a release of a red coral larva. So all this knowledge, of the starting knowledge, is useful because we can answer to specific scientific questions. One of that is the fertilization distance. That will be the topic of Bruna and the PhD of Bruna Giordano, which we speak later. Then we can test the hypothesis on spatial distribution of the settlers and survival after implantation, meaning intraspecific competition. And we can even study larval settlement preference, biological, physical, and microbial. So this is clearly a laboratory. And in conclusion, we have a species for which passive restoration is ongoing. So in some places, the disturbance are eliminated. So the active restoration is needed and it can be done. And we wonder if transplantation or sexual restoration of a mix of the two techniques is the best one. And to understand this, we create an artificial population in an underwater laboratory to apply a scientific approach to restoration with the final goal. So it's not really a coraliculture, but it's a laboratory in which the final goal is develop a scientific protocol for coral local restoration or restocking and breeding. And this project was funded by Fondation Prince de Monaco. So I thank you everyone for your attention. And if you have a question, probably there's still a little bit of time, but probably later. Thank you.
Thank you, Lorenzo, for explaining so well all this, giving all this information, and that allow now to listen to Stephanie. We will give you concrete examples of what we do with these caves in Monaco. Stephanie, please. Stéphanie Reynaud is a senior scientist at the Centre Scientifique de Monaco in the team of ecophysiology.
So good afternoon. So I represent now you the newly born Corallium rubrum colonies in Monaco caves. As Lorenzo already told you, passive protected area is insufficient for recovery the very degraded habitats. So basic transplantation techniques are completely not appropriate for a Corallium rubrum because this is a very extremely slow growing species. And the only way to obtain more colonies is the reproduction and generation of new colonies. So to this aim, we set up colonies of Corallium rubrum in artificial caves and focus on finding the ideal sex ratio, the ideal substrate, and the ideal colony density to facilitate an optimal reproduction. So these caves constitute an underwater laboratory in semi-controlled conditions. So in May 2021 in Monaco, we built some artificial caves equipped with rails of the roof to hold plates supporting red coral colonies. And these structures were submerged in front of Monaco at 40-meter depth. You can see here the location chosen in front of the harbour, and the yellow triangle is the Centre Scientifique de Monaco, so very close to the caves. And the boat is the yellow rectangle. So the caves were put in hexagonal shapes, as you can see. At the beginning, they were empty of corals. And in June 2021, the red coral colonies were sampled off the coast of Nice at 50-meter depth and placed in the aquarium of the laboratory. The sex of each colony has been determined, and we have obtained 31 females and 33 males, so the balanced sex ratios.
Monseigneur, Excellency, dear guests, thank you very much for attending our meeting. And you are just in the middle of the talk, which is about the caves in Monaco.
So 4 tiles have been glued on each of the plates, 2 in terracotta, 1 brown and 1 red, and 2 in marble, white and red. And the small colony of red coral have been placed in the middle of this plate. In July 2021, the plates have been distributed into the 6 caves, as you can see of these pictures. The sliding tiles can be removed, and we can change the order, the sex and the female. Since the beginning of the experiment, we have monitored the temperature in front of the caves, and you can see that in the winter, the temperature reaches 14 degrees, and can reach in summer 24 degrees, but only during few hours. The current was also monitored, and you can see that it's very different during the year, and I remember that the spawning is in July. In the first summer, in 2022, little or no reproduction took place, and 2 hypotheses were considered. The colony maybe were not yet adapted to their new growth condition, or the coral biomass in the caves was too low to maximize the encounter of male and female gametes. So therefore, before the next summer, the number of male and female colonies in 2 caves was increased, while maintaining the same sex ratio, as you can see here. So in the cave 5 and 6, we increased the biomass of the corals, and the sex ratio was identical as before. And for example, in this cave, we have 5 female for 1 male, 3 female for, and so the sex ratio was different in each cave, and we wanted to find the ideal sex ratio and colony density. This year, in January, all plates were brought back to the laboratory to quantify larval recruitment. And for example, on this plate, on the cave 1, we obtained a total of 33 recruits, 5 on brown terracotta, 4 on red terracotta, 12 in red marble, 6 in white marble, and also 6 in the plastic white substrate. Another example of the distribution, we have obtained 33 recruits on white marbles. You can see here a picture of a recruit about 2.5 millimeter, that is more or less 2 or 3 years old. So the results, at the end, we have obtained a total of 241 recruits on all the plates. The distribution on the marble was 143, and on terracotta 84. So in conclusion, I can say that coral colony density does not appear to affect the number of recruits obtained. However, the sex ratio plays a crucial role, with the imbalance favoring female colonies being a significant determinant. Additionally, the direction of the current is another important factor that must be considered. And finally, the recruits show a clear preference for marble as a substrate for settlement. Thank you for your attention.
Thank you for this presentation. And so we start to see how to have the best condition for growing these corals. And we have baby corals just in the cave in front of Monaco. And now we are going to listen to Bruna Giordano, who is the PhD student of Dr. Bramanti, and she will explain the results they have obtained in Bagnols-sur-Mer. Please.
Thank you, everybody. Thank you, Your Majesty and colleagues. I'm Bruna Giordano. I worked with Lorenzo Bramanti during my PhD that I've just finished these days. And today I would like to, I had the pleasure to work in this nice project about red coral restoration, in particular in Bagnols-sur-Mer, but with the collaboration of Centre Scientifique de Monaco. So today I would like to present to you the result of one of the studies that I did during my PhD. In particular, I aim to understand the fertilization mechanisms of Mediterranean red coral in order to apply this information to transplantation protocols. So we tried to answer particular 3 questions. The first one, it was what is the maximum fertilization distance that red coral can achieve? The second one, it was, is it possible to observe an effect of sperm dilution on the fertilization success of female colonies in Mediterranean red coral? And the third one was if sperm limitation could affect female larval production in this species. In particular, to make a brief introduction about this subject, we know that in natural endangered population that no decreasing, can be especially vulnerable to the Ali effect. The Ali effect is a phenomenon where the individual fitness or population growth can be declines when population density is too low. In particular, this population that know declines under a certain threshold may face challenges in, for example, finding mates, or can suffer by reduced genetic diversity. What I did? Okay. Due to inbreeding depression. So for this reason, coral restoration must account for population thresholds and genetical features such individual distribution and density, which are crucial to ensure population successful reproduction over time and to ensure long-term survival. So in this context, I would like to introduce 2 main terminology. What is sperm dilution? Sperm dilution is the decrease of sperm concentration in the environment, which can reduce the probability of sperm to successfully fertilize eggs, and sperm limitation is its direct consequence. And it occurs when the reproductive success of females is restricted due to insufficient sperm concentration in the water column. Okay, sorry. So how Lorenzo Bramanti explained before, trying to answer this question by studying natural population could be extremely challenging because it could require a huge sampling effort. For this reason, we established an artificial coral population thanks to this system of artificial caves. In particular, during this experiment, I was interested in regulating 2 main parameters. The first one, it was the distance between the caves in order to study the sperm dilution, the effect of sperm dilution on the fertilization of red coral colony. And the second one was the sex ratio within the caves that allowed me to understand the effect of sperm limitation on the female larval production. Okay, so to answer this question, we took advantage of molecular tools, in particular to perform a parentage assignment. So during 2 reproductive seasons, we collected all the larvae produced by the females colony that were placed before in the system of artificial caves, and we genotyped both larvae and parents in order to perform parentage assignment. So in these figures we can see like the pattern of parental assignment. Each point Each point represents the number of larvae produced by a female colony, which is represented as a function of the distance from the siring males. So from this graph we can keep two main information. The first one is that there is a significant effect of sperm dilution on female fertilization, in particular males that are placed in the same cave than females, so at close distance, at less than 1 meter square distance from this female, have a higher fertilization success than males that are far from females. And the second result indicates here that maximum fertilization distance in these species, surprisingly to respect to the literature, can reach even 13 meters. To address the final question of the experiment, I illustrate here the impact of male presence or absence on female fecundity. So we arrange with different sex ratio the corals in the system of artificial caves, and in this graph we can see that females that are placed in the same caves that male show a significantly higher larval production than females that were far from males, that were isolated from males, showing that there is an effect of sperm limitation on larval production. These results allow us to take some messages home, in particular that there is the significant effect of sperm limitation and dilution on the fertilization dynamics of Mediterranean red coral. Second, that there is, and this information is crucial when we need to plan any restoration and transplantation plan. In particular, it is important to recreate and restore functional patches capable of sustaining breeding interaction between colonies. In particular, we suggest to create small, dense patches of reproductive colonies within a small area of half meter, 1 meter square. So thank you for your attention.
Thank you, Bruna. So now we have listened to 3 talks with information obtained in the field, and we are now going to move to a presentation by Dr. Philippe Gannot from the Centre Scientifique de Monaco, and he will present the results that we have in the lab concerning growth of Corallium reborn.
Monseigneur, dear colleagues, thank you very much. So we're going to change a bit the gear and go into, now we're going to shift the culture and go what happened into the lab. So I will start to quote a well-known sentence: you cannot protect what you don't know. And if you want to know more about the red coral, you need to study its biology, but at the level of fundamental research. And to do so, you need to have on-demand, reliable material that you provide with the culture, where you have both control condition and monoculture. And at the CSM, we have been developing 2 types of cultivation, I would say, either by hanging or by lateral growth. So we started in 2013, and we picked up colonies. The first one was in Marseille, so we always pick up colonies like just branches. We don't pick up the whole colony, and they make between 5 and 10 centimeters. What you have on the left hand is the first sampling, where you see the branch that they are hanging on just nylon strings. And through the time, we've been renewing our culture, and with the increased demand of the CSM Channel program, we've been increasing the number of samplings. And typically now we have over 200 colonies that are now cultured on hanging rods, rigid, that limits the fluctuation in between the colonies, and just the colony are glued onto the stick and hanging into the water. One way to measure the evolution of colonies is to measure the buoyant weight, is that you put them into the water and you measure in grams what happens through the times. So that's the graphic you have on the right side with the days. And you can see that we have different response. We have some of them that are fast growing, like having increase of mass rapidly, and some others that are slow growing. But on the picture on the left, I showed you our champion. So the colony in A is typically, sorry, what would happen to a colony just take out from the field. And this is a colony that has 8 years of culture, and you can see that it grew immensely compared to the others. So what we have is a much faster growth. Then we wanted to have more resolution on what happens to the growth, and we developed the 3D scanning of living colonies, so the colonies are not dead. So what you have on the right side is our engineer who is making the scan with a laser scanner. And as you can see, if I take, for example, a colony on September 2022, this is how it looks like, and after 3 years, or this is a 3-year culture, you can see that there is much more ramification. So by having the 3D structure, you can already see that one of the way of growth of our colonies is that it substantially ramifies. Then, if you look 2 years later, and you continue monitoring the same colonies, you see that they grew again. And so this should be rotating, but I could not have it. So if you look at the colony on top, the colony called IAL, it grew by 250% in terms of volume, whereas the second one just grew by 56% of the volume. And what you can see is that it did not increase by length of the branch, but more by the ramification. Another information we can have from the volume is that we can calculate the density, and we can see that the density is decreasing, which is indicating that in fact we have, that's how we interpret the system, is that we have more tissue growth than calcification. Tissue are almost buoyant, whereas the skeleton is heavy. We call those morphotypes, we call them sumo because they have an appearance like me. And this is another illustration of how it grows. So this was the colony just before. This is 2022. If you superimpose what happens after 2 years, you can see that it grows just by increasing the size of their branches. And therefore, we can follow the special growth of evolution without killing the colonies on these techniques. So these are our models. They are aquarium colonies. Of course, we don't use them just for the sake of growing them. We do use them through our different techniques, such as microscopy. We use biochemistry, bioinformatics, and that allows us to study different factors like growth control, tissue growth, skeleton, on the color, just from the tissue to the organism, back to the molecular. Another aspect that we have developed is the colony that we call lateral growth. So this was developed in 2017 in the lab, inspired from what was already known on tropical coral. So what you take is you take an apex of a branch, very small, just at the tip, and you glue it on the slide. And using the right condition, what you will see throughout time, this is the pictures just on the bottom, is that it can grow the tissue on the slide instead of growing like a tree. And with time, you can see that the tissue grows on the slide, calcify and reproduce all the parameters of the living colony, meaning that we have exactly the same type of growth than if you had branches, but they are easier to visualize. I will show you later. And now we cultivate them on the tower. We call them the colony towers. You have pictures of them outside in much broader. And again, if we look at the expansion, the difficulty, in fact, for this type of culture is to start the growth. But once they start, they can grow rather fast. And the idea is that once it's started, the expansion is more or less steady. And this is very nice material. So nowadays we have 50 colonies growing like that. You see the picture on the left is an aquarium. And you can see how the colony on the right can be envisaged. And you can even make time-lapse video of them, and that way, what you can see, it should be moving, but that's the video. No, it's moving, okay. And you can see, you can follow the variety of the polyp, so you can study either the tissue growth, either the polyp, the canals, or even the calcification processes, just at that level that's much deeper than what you can do without. So the take-home message is that the culture of the red coral is an essential element that we must master if you want to propagate it. That is the message. As a conclusion of what I just showed you, I would tell you that we've been keeping colonies for more than 10 years now, and that we succeed into that. I think we are the only one in the world who managed this type of growth. That we have a growth rate that are high, I would say much higher than in the nature, although they are variable, and this is likely reflecting the genetic variability in between individuals. and that these are really biological materials, for our fundamental research. If we will look into further prospects, there are two things that we don't have. The first one is we don't control sexual reproduction. So if we need to have larvae, we have to go back to the sea, like was presented before. And the second thing that this laboratory culture could serve as sources for reintroduction program aimed at enhancing the natural population as it's here. Thank you very much.
Thank you. So you can see that we have now information not only in the field but also in the lab, and we learn more and more about the way to boost coral growth and maintain it in culture. And now we are going to listen to Dr. Romy Tigna-Perrier from the Centre Scientifique de Monaco, and she will discuss about the microbiome of the coral.
Son Altesse, distinguished guests, colleagues, organizers, good evening. Thank you for inviting us. I'm really happy to be here and be able to present our research on the microbiome of the red coral. It is now commonly accepted that the microbiome is really important in the functioning and the health of all animals. Human is often taken as an example, as it is the most studied model of symbiosis between an animal host and its microbiome. The microbiome is composed of all the microorganisms that are living on and within the animal host, and it has diverse functions in humans, including in the nutrition, immune system, and resistance to stress, and protection against pathogens. So, because of its high importance in the human health, the microbiome starts to be used to do personalized medicines, to do disease prevention, disease diagnosis, and disease treatment. And because there is no exception, the red coral is also associated with a bunch of microorganisms that are important to investigate to better understand the biology of Corallium rubrum. So I'm going to focus my talk on the bacteria that our team has studied the most, but there are also other microorganisms named microeukaryotes that are fungi, protists, algae that are associated also with the red coral, but I'm not going to talk about them. So we show that the red coral is associated with a specific bacterial microbiome dominated by a bacterium, the Spirochaetaceae, that we can find always in association with red coral colonies, whatever the sampling time, the sampling depth, or the sampling place in the Mediterranean Sea. So, given its high stability, we wondered how this microbiome, especially these Spirochaetaceae bacteria, are acquired by the red coral, and if it was through a mother transmission. So to do so, we followed the microbiome composition from newly born larvae of different ages, settlers, recruit juveniles, and we show that the Spirochaetaceae are acquired from the environment, so not from the mother colony, and during the first years of life of the red coral when it's still a juvenile. So the Spirochaetaceae bacteria don't seem to have a role in the ontogeny of the red coral, but our metatranscriptomic analysis that allows to analyze all the microbial functional genes expressed show that these Spirochaetaceae bacteria might have a role in the nutrition of the red coral, and especially by synthesizing essential vitamins and amino acids that the animal cannot synthesize. Part of our research focuses on the response of the microbiome to environmental change, such as marine heat waves, and the role of its microbiome in the resistance of the red coral to heat stress. We know that the red coral starts to die below 23 degrees Celsius. That is a temperature always reached during marine heat waves in the photic zone of the Mediterranean Sea. And these marine heat waves cause mass mortality events. For example, in 2022, we observed up to 80% of the colonies affected by tissue loss, depending on the site in the Mediterranean Sea. So based on lab experiments and in situ measurements during the 2022 marine heat waves, where we collected a lot of samples of different health status, we found that the proliferation of microbial opportunists and putative pathogens contribute to the mortality of the red coral during marine heat waves. Surprisingly, we found that the Spirocataceae seem not affected by the marine heat waves, as nor their abundance nor neither their functions were altered during the stress. So it seems that Corallium rubrum continues to benefit from the nutritional support of the Spirocataceae bacteria, even under abnormally hot temperatures. I would like to thank you.
Thank you, Romy, for showing us that not only for humans but also for the corals, the microbiome is so important. And we should have had a talk by Professor Maria Concetta Di Natale dell'Università di Palermo, but unfortunately she couldn't come because of last-minute obligation, so we are going to move directly to the movie by Roberto Rinaldi. Roberto, please, can you come? Oh, there is one. Do you want to say a few words presenting your movie? Yeah, thank you.
Good morning, good evening. Just a few words, not so much to say. I mean, my job is to be a documentarist, always working on the sea, mostly under the sea, underwater. And so it is now a big passion to try to divulgate what the researchers and the scientists are doing. I grew up as a professional with the Commandant Cousteau. I was on board of Calypso so many years, so I was also living Monaco in the former times. And the best thing that he taught us that everybody is protecting what he loves, but nobody can love whatever doesn't know. So we have to know how beautiful is our sea, our ocean, and how beautiful is protecting. And I believe that following the scientists in some work like this one is passionating, because you see the strength they put on, the passion they put on, and becomes really not to tell the story of a research, but rather to tell the story of an adventure. And then with my collaborators, with Manuela, with Marco, we try to put together, yes, laboratory interviews, and then all our knowledge about the sea, the Mediterranean Sea, going down in the water and trying to find the most beautiful place where coral is still growing in nature. And I mean, the two things are matching together, because these people is doing a big strength to bring the Mediterranean back to the status where it was. Thank you.
corail rouge est une espèce emblématique de notre mer. Éclatant de rouge, solide, exploitable, il a été pendant des siècles l'objet de chalutage, puis du braconnage et des effets du changement climatique.
Malgré les nombreuses mesures de gestion appliquées au niveau de l'interbassine méditerranée, avec l'objectif de réduire le déclin de ces populations, la croissance lente de cette espèce nous suggère l'utilité des mesures de restauration active, surtout dans les zones où la pression de la pêche a été éliminée, comme par exemple les aires marines protégées.
La restauration des écosystèmes est une branche de l'écologie de la conservation. C'est une science jeune, particulièrement pour le milieu marin, qui concerne surtout les récifs coralliens. Mais ici à Monaco, nous nous sommes intéressés à la culture du corail rouge depuis plus de 30 ans. En effet, c'est dans les années 1980 que nous avons imaginé la construction de grottes en béton de 13 mètres cubes afin de pouvoir faire vivre, croître et se reproduire le corail rouge avant de le réintroduire dans le milieu naturel.
Dans les années 80, j'étais une étudiante de biologie et j'ai creusé les travaux et les recherches de Denis. 30 années plus tard, j'ai l'occasion de donner ma contribution. Sur la base des grottes de Denis, j'ai conçu un système, un véritable laboratoire sous-marin où les colonies de corail rouge pouvaient être étudiées et élevées dans des conditions naturelles.
Aujourd'hui, l'objectif des collaborateurs de ce projet, le Centre scientifique de Monaco, le CNRS, l'Observatoire de Banyuls, supporté par nos sponsors, la Maison Chanel et la Fondation Prince Albert II, est encore plus ambitieux. C'est d'essayer de mettre en place une reproduction contrôlée du corail rouge afin d'augmenter son succès reproductif.
Les coraux se reproduisent par l'émission de larves. Ces larves, d'environ 1 mm de long, sont libérées une fois par an pendant la période estivale et voyagent, transportées par le courant, pendant environ 20 jours. Ensuite, elles se posent sur un fond adapté, se transforment en un petit polype de corail et à partir de ce moment, pendant une centaine d'années suivantes, elles vivront attachées au substrat.
C'est une bestiole bizarre, le corail. Pendant les premiers 20 jours de sa vie, ils vivent dans les courantes marines, dans la colonne d'eau transportée par la courante marine. Et après, pendant les suivantes centaines d'années, ils vivent fixes au substrat.
Les premiers 20 jours sont très dangereux. Les minuscules larves sont des proies recherchées par des centaines d'espèces de poissons. Et en fait, seule une toute petite proportion survit. La plupart meurent avant même de pouvoir devenir un corail.
Voici l'intuition qui a dirigé notre projet. L'idée, c'était de réduire la mortalité drastique des larves qui sont émises dans le milieu extérieur, proche de 100%, afin d'augmenter le succès de la reproduction. À cette fin, les grottes ont été conçues de manière à ce que les larves produites à l'intérieur de la grotte restent piégées dans la cavité, afin de réduire la prédation sur celle-ci et d'augmenter ainsi fortement la capacité de reproduction du corail rouge.
Une deuxième stratégie, en cours de vérification, c'est basée sur une caractéristique péculière de ce nouvel système de grottes, où les colonies peuvent être extraites et réinsérées facilement. De cette façon, les mères en train de libérer des larves peuvent être installées dans un aquarium et élevées dans les meilleures conditions.
Les bébés coraux qui sont nés en aquarium sont maintenus dans des situations de sécurité jusqu'à l'âge de 2 ans, quand ils arrivent à former un squelette calcaire. À ce moment, on les transplante en mer, soit sur un substrat rocheux naturel, soit dans les grottes artificielles qu'on utilise pour nos études et nos recherches.
Un jardin de corail qui ne provient pas du prélèvement dans la nature de certaines colonies, qui sont ensuite réintroduites ailleurs, mais d'un véritable élevage de corail. Où les chercheurs tentent de percer le mystère de la reproduction et d'élever les larves jusqu'à ce qu'elles se transforment en individus fixés sur un substrat réintroduisible dans les fonds marins. Dans le domaine scientifique, des dizaines d'expériences de prélèvement de branches de coraux dans la nature et de réimplantation dans des zones où ces espèces étaient en difficulté ont été réalisées. Beaucoup n'ont pas pris en compte les problèmes fondamentaux, compromettant ainsi complètement le résultat escompté. Le premier d'entre eux est la compréhension de la biologie reproductive de l'espèce.
Dans le corail rouge, il y a des mâles et des femelles. Et la détermination du sexe est fondamentale pour les plans d'élevage. Imagine une ferme où tu veux élever des vaches et tu ne sais pas la différence entre vaches et taureaux. Ça va être une catastrophe.
Pour cette nouvelle expérience, en plus de continuer à capter les larves à l'intérieur de la grotte, nous avons déterminé le sexe de chacune des colonies afin de calculer quelle était la meilleure combinaison de mâles et de femelles à l'intérieur de la grotte. Et au-delà du rapport mâle et femelle, nous cherchons également à comprendre quelle est la distance à laquelle les mâles sont capables de féconder les colonies femelles. Et le deuxième était quel est le nombre de larves que peut émettre une femelle.
Les réponses à ces questions viennent de la génétique, qui est en science moderne sur laquelle s'appuie une grande partie de l'intérêt de notre projet. Des informations fondamentales sont contenues dans l'ADN, comme par exemple le sexe du coraux, ou sinon aussi le degré de parenté entre le bébé et les parents coraux. Les laboratoires équipés et les aquariums sont des véritables services de maternité où les coraux sont transportés des grottes artificielles en mer jusqu'aux aquariums où ils peuvent se reproduire et pondre des larves qui sont suivies jusqu'à la métamorphose et à la fixation sur des substrats qui sont ensuite transportés de nouveau en mer, en milieu naturel ou de nouveau dans les grottes artificielles.
Aujourd'hui, le Centre scientifique de Monaco est devenu spécialisé dans le contrôle de la reproduction du corail rouge et dans l'étude du développement depuis le stade larvaire jusqu'au stade polype. Et ceci pour assurer un meilleur succès reproductif. Pour cela, les chercheurs du Centre scientifique de Monaco ont établi des stratégies et aidé des outils de pointe, espèrent aboutir à des résultats qui sont fabuleux.
Pour notre projet, on a besoin des exemplaires de coraux, mais il est un dommage de les extirper dans son environnement naturel. Du coup, on se trouve souvent en face d'un dilemme. Mais c'est la mer, par défaut, qui nous offre des solutions inespérées. Et nageant dans les eaux de Bagnols, sur le fond de Bagnols-sur-Mer, je trouve des colonies de corail rouge détachées, destinées à mourir se lasser sur le fond. Ces colonies sont pour nous une excellente occasion d'étude et une source de larves vers notre élevage. Mais comme ces branches sont tombées, qui l'a cassé dans une des réserves marines plus contrôlées de France? C'est pas que l'homme qui peut casser les colonies de coraux. Ici, un possible responsable. Aussi, un mérou qui rentre dans son trou peut être le coupable.
L'hôtel Corail, ainsi a été surnommé le système de grottes artificielles, est un véritable laboratoire sous-marin axé sur l'étude des stratégies de reproduction du corail rouge et sur le développement d'un protocole de réintroduction dans la nature basé sur les caractéristiques biologiques de l'espèce. Conformément au principe des solutions basées sur la nature, nous orientons toutes nos actions dans le sens de ce qui se passe normalement dans la nature.
Observer la morphologie des fonds marines, comprendre con quels critères les colonies se distribuent dans l'espace, comprendre l'alternance entre lumière et sombre, comment utilisent les courantes. C'est le travail des naturalistes qui nous inspire pour la conception des systèmes de laboratoire sous-marin. Cette grosse sous-marine en fond de sable, avec la veau riche en coraux rouges, c'est pour moi une inspiration. Je l'ai étudié beaucoup de fois, pendant beaucoup de temps, pour comprendre quelles sont les préférences des larves pour se fixer, quelle est la distribution et la différence de distribution spatiale entre les individus femelles et mâles. L'observation de l'architecture naturelle, c'est l'inspiration pour la conception des grottes artificielles.
Bonjour à tous, on est là pour présenter les résultats de l'expérience de corail-culture in situ que nous avons réalisée à Monaco. Et je crois que les résultats ont dépassé toutes nos attentes. Lorenzo, tu peux nous en dire quelques mots?
Ah sì, per rapport à la settlement, les larves qu'on a obtenues se sont fixées sur les substrats qu'on avait préparé sur les aquariums et on a vu un taux de survie qui était 10 fois plus grand, supérieur comparé avec les milieux naturels.
En effet, donc en 2023, on a eu plus de 250 larves fixées sur ces substrats, donc des larves nées à Monaco, donc ces grottes expérimentales immergées ici dans les eaux monégasques. Maintenant, il va falloir commencer à étudier, savoir pourquoi ces larves ont choisi certains substrats par rapport à d'autres et voir comment elles contrôlent leur croissance durant leurs premiers jours de vie fixés.
On a beaucoup à travailler encore.
Tout à fait.
Ces résultats sont essentiels pour l'élaboration des plans de restauration active basés sur des connaissances scientifiques. Mais tous les plans de restauration active, il n'y a pas de sens s'ils ne sont pas précédés d'un plan de restauration passive, c'est-à-dire l'élimination ou la mitigation des conditions qui ont généré les problèmes. C'est comme si on veut planter des arbres dans une forêt qui encore brûle.
Il faut bien avoir en tête que les zones marines protégées jouent un rôle fondamental en fournissant des environnements où les pressions anthropiques sont minimisées et où les conditions écologiques sont favorables à la récupération des espèces.
C'est de là que nous commençons à défendre la mer pour sauver ses habitants. Un univers magnifique et complexe. Un monde extraordinaire, riche en vie. Contrairement à la terre ferme, il est réparti dans les 3 dimensions. Rochers, falaises, grottes, étendues de sable, immenses espaces d'eau libre constituent un monde encore largement inconnu pour nous.
Même imaginer qu'après des années de souffrance, les hôtels à corail mis au point ici à Monaco et à la station de Bagnouls puissent être utilisés de partout en Méditerranée pour repeupler les populations de corail rouge.
Thank you, Roberto, for this movie that illustrates so well our research program. I don't know if we have time for questions, I'm not sure. Laura, maybe you want to come with us to say a few words?
Merci Sylvie, merci le groupe des scientifiques. Vous nous avez fait rêver un monde de magie où la chimie parle, où on revient aux origines et on comprend encore plus pourquoi ces espèces sont tellement fragiles. Chaque élément s'ajoute à la liste de risques qui peuvent interrompre ce fameux cycle qui a l'air tellement vulnérable. Donc je vous félicite, on n'a pas arrêté de parler de vous, ça fait déjà 5 jours que pendant le congrès, on vous attend parce que l'exposition qui vous avait été dédiée est encore là, mais on voulait savoir justement s'il y a de l'espoir pour ces coraux rouges tellement abîmés par nos propres erreurs. Je ne sais pas si vous voulez nous donner quelques mots à ce sujet. Mais je vais juste entendre des bonnes nouvelles, s'il vous plaît.
Oui, merci. Moi, je pense que le fait même qu'on s'engage dans des projets de restauration signifie que l'espoir est là. Et... Ah, sorry. So the fact that we start this program of restoration, it means that we have we have espoir. I shift to phrase. So we have we can hope. Yes, sorry. So we have hope that something good can happen because if not, we don't apply this. The message is that sometimes this hope is brought by scientists. We see a lot of program of restoration that is based on cut and paste, and the message that we want to bring here, that we try to put at the base of the hope the scientific approach. So we hope that we can take the lead of this restoration of the corals, just putting what we know to do better. So applying question, posing question, and trying to answer to these questions.
I will add something even more positive, referring to what was just before with the deep sea. These animals are living very deep, and as much as they have suffered of anthropic pressure on the first 100 meters below the population is, I would not say unaffected, but they are there.
Thank you very much. That's really, this part of science is really so much useful for us to imagine that despite of all of the mistakes that we have made, we can start again to build. So I really thank you all for being there. I don't know if there is just time for one question from the floor because we are really right in time. So we have the cocktail this evening and I think everybody wants to have a break before. But if the public has some...
Thank you. Yes, it was a great presentation. Not a scientific question, more I was puzzled about the substrate that they prefer, marble compared to terracotta. Why did you choose this and how this can relate to what could be found in the natural environment? And yes, and why more luxurious substrates? I don't know.
Well, at the beginning, we don't really know what kind of substrate we can use. And because we have several clues for your studies, we think that terracotta could be the best. And we had marble, we don't know, like this. And in fact, they prefer marble because they are monegasque.
By the way, my origin are from between Tuscany and Liguria, so I live close to Carrara. And so my PhD thesis was done using marble, the purest marble in the world, that is Carrara 1, and red coral used to like it. So we started from this like an hypothesis, but the fact is that Romy, better than me, can explain probably the response is the bacterial and microbial community, not just in the substrate.
Yes, you want me to talk about the...
I can say some words also.
Just maybe Romy confirmed that the substrate is not only important because of its composition, but because its composition and the way the coral interacts with it is through bacteria and other.
I think the substrates are different physically. You have, I don't know, you have structures or not on the substrates, but you have also different biofilms on the substrates that can maybe attract the larvae or not. So this is also one question we would like to answer, and it's for the future, but we don't have an answer yet.
Thank you, it's fascinating, thank you.
I have just an addition, just because it was my ancient passion to study about the interaction of microorganisms, etc., and then biotechnology. You know why the shark is not anymore so much aggressed by biofilm is because of the nanometer structure of the surface. So it might be, maybe, that the structure at this level of porosity is ideal for the dimensional larvae. Sometimes it's just something like this, but I don't know if it is the case.
It could be, this is something that we are looking at. Yeah.
I thank you very much, really. It was a pleasure to have it with us, and I hope you are going to enjoy a little bit what the rest of the Congress. And
yes, thank you so much for inviting us, and thank you so much for giving us the opportunity to present you this research program that I think also is full of hope. Thank you.